If your eyes are open at 1:17 in the morning, listen closely. Earth is still hiding whole chapters from science, and tonight we will drift through 13 gentle mysteries. We will climb an evidence ladder each time, from observation to measurements to ideas, and end on the one detail that still will not settle. If it helps you settle, like, and save it, then comment one word: ocean, sky, animals, or rocks. If you have watched a bird slip into dusk, you have seen a compass at work, even though there is no needle to follow. Somewhere inside that small body, direction becomes a steady feeling, like a quiet pull toward a distant place. Our first mystery is simple to name and still hard to finish. How do animals sense where they are going without a map? We will keep a soft loop open, because later we will meet rare lights in the sky that also depend on invisible fields. Across the planet, many travelers repeat routes with calm precision, year after year, even when weather and coastlines shift. Songbirds cross continents, sea turtles cross open ocean, salmon return to home rivers, and bees return to the same hidden flowers. The puzzle is not only success, but similarity, because very different bodies and brains can reach such steady accuracy. A turtle hatchling has no teacher, yet it begins a journey that seems to fit an ancient path. A young bird can leave on its first migration and still choose a season and direction that makes sense. When navigation works, it moves seeds and nutrients and life across regions, holding ecosystems together in a gentle rhythm. When navigation fails, the ripple can be wide, from missed breeding to changed feeding grounds and altered food webs. So researchers ask one calm question from many angles. What cues do animals use, and which cue matters most when conditions change? Some species use the sun because its daily arc carries time and direction together in a simple pattern. In clock shift studies, when an animal's day is nudged earlier or later with controlled light, its sun-based heading can shift too. Others use the stars, because night patterns can stay stable when the ground below offers little guidance. In planetarium tests, changing the apparent rotation of the night sky can change a young bird's chosen direction, as if the sky teaches a reference. Many use smell because air and water carry chemical signatures that form a living landscape close to home. And many use landmarks, like coastlines and valleys and river bends, when those shapes are available and reliable. Then there is magnetism, a planetary feature you cannot see, yet one that wraps Earth like a soft, steady shell. The magnetic field has strength, and it also has tilt, and both properties change with latitude. A key detail is that many birds seem to follow tilt more than polarity, so they can use an inclination compass rather than a simple north detector. Near the equator, the field is more level, and toward the poles it leans, like a gentle slope you cannot touch. If an animal senses strength and tilt, it may gain a rough position as well as a heading, like knowing where you are on a hill. That possibility, a magnetic map, is why this mystery stays so compelling and so carefully tested. Our evidence ladder starts with observation, and the observations are abundant, from repeated timing to stable migration corridors. Modern tracking tags add clarity because tiny loggers can reveal routes that were once invisible to people on the ground. Some tags record light levels to estimate position, while others talk to satellites, and together they show how steady many routes remain. Many animals follow corridors that persist across years, even over open water where landmarks are scarce and winds can be strong. The next rung is controlled testing, where one cue is changed while the animal is otherwise kept safe and calm. For migratory birds, researchers use orientation cages, where a bird expresses its preferred heading through gentle movement. When the surrounding magnetic field is rotated using wire coils, the bird's heading often rotates too. In some field studies, adding a small magnet can increase scatter, while leaving a non-magnetic weight does not, though results vary by species. That is a clean clue, because it suggests the animal is responding to a field that has been quietly altered. In other tests, magnetic strength and tilt are shifted, and some species respond as if they notice that change as meaningful information. Sea turtle hatchlings can be tested in calm tanks, where they swim as if beginning their first ocean phase. When the magnetic field is set to match values from different parts of the natural route, hatchlings often change course in the expected direction. This has led to the idea of magnetic imprinting, where early life stages learn the magnetic signature of key places along a journey. It is as if the hatchling recognizes a magnetic neighborhood and chooses a safer heading, even with no past experience. Similar patterns appear in fish, where magnetic cues may help during long ocean phases when smell and landmarks are limited. All of this suggests the sense is not just north and south, but includes tilt and strength, which is a richer kind of information. Now we add the limits, because the same effect can weaken in labs and not always for the same reason. Bright artificial light, unusual wavelengths, and the stress of captivity can reduce the clarity of magnetic orientation. Some labs also report that faint radio noise from electronics can interfere, which is surprising and still debated. If a compass depends on delicate chemistry, then small artificial signals could matter, and that possibility is now part of careful lab design. This mismatch matters because a sense that works outdoors can become difficult to capture indoors on demand with perfect repeatability. So we move to the leading hypotheses, which try to connect a physical sensor to a nerve signal that the brain can use. The first major candidate is magnetite, a tiny iron-based mineral that can behave like a microscopic compass inside tissue. If magnetite crystals sit in the right place, a magnetic field could nudge them, and that motion could activate nearby nerves. Researchers have searched for iron-rich structures in birds and fish, hoping for a clear organ devoted to magnetism. In a few cases, structures near the inner ear have become candidates too, because balance organs already detect tiny forces with high sensitivity. Some studies have reported promising cells near sensory nerves, but others argue that certain iron deposits may have ordinary immune roles. So magnetite remains plausible, but it has not become a single universal answer that fits every traveler. The second major candidate begins with chemistry in a process called a radical pair triggered by light. In some animals, light-sensitive proteins in the eye, such as cryptochrome, may create brief chemical pairs whose behavior depends on magnetic fields. Under this view, the animal may see a faint internal pattern, like soft shading that shifts with direction. People sometimes call this quantum-like because electron spins follow rules that differ from everyday objects you can hold. Support comes from studies where some birds orient well only under certain dim light conditions and lose orientation under others. That light dependence is one reason the eye-based hypothesis stays strong, even though the exact location of the sensor is still argued. In some experiments, weak oscillating fields can disrupt orientation, which fits parts of the radical pair model. Still, the details are hard. Because the signals are tiny, the chemistry is fast, and living tissue is warm and noisy. It is also possible that animals mix strategies using chemical sensing for direction and magnetite for map information, or switching as needed. This multi-compass idea feels natural because travelers often keep backups using the sun, the stars, smells, and magnetism together. Young animals add another clue because many show useful headings before learning the route, hinting at built-in programs. Over time, experience can refine the path, and calibration can align magnetic cues with sunsets and star patterns. So the web of evidence is real. But the final chain is still missing, from sensor to brain map to the exact turn taken in the wild. Part of the challenge is faintness, because the magnetic field is gentle, and the biological response may sit near the edge of detection. Another challenge is diversity, because different species may solve the same navigation problem in different ways. To move forward, scientists build better tags that record location alongside local magnetic values, wind, and light for clearer comparisons. Laboratories also improve shielding, reducing stray electromagnetic noise, so magnetic responses can be tested more consistently. On the biology side, researchers look for genes and proteins that track with magnetic behavior, and for signals that can be measured in living tissue. In some insects and small lab animals, Changing light-sensitive proteins can change magnetic-related behavior, hinting that the pathway is real. Translating that kind of test to long-lived migrants is slower, but the direction of research is steady and patient. So here is the gentle takeaway. Many animals use multiple cues, and magnetic information appears to be one of them. The leading ideas include magnetite crystals and light-triggered radical pair chemistry, and perhaps a careful blend of both. What still will not settle is the exact sensor in each species and how the brain turns a faint hint into a reliable map. And we will return to invisible fields later, high above the clouds, in quiet forms that science is still learning to measure. Now, from a hidden compass, we move to an animal that travels far, yet hides its most important destination. Picture a quiet river at night, where the surface barely moves and the banks feel asleep. Somewhere under that calm water, an eel is preparing for a trip it may never return from. This is a migration with a missing ending because the final act happens far out at sea, mostly unseen. We will keep a soft loop open here because later mysteries also depend on subtle cues across wide distances. European eels and American eels can live for years in rivers and wetlands, growing slowly in the dark. Then, at a certain season and size, something shifts and the body begins to change for travel. The change is called silvering, and it is less like a costume and more like a full retooling. The skin becomes brighter and more reflective, which may help in open water and deeper light. The eyes enlarge, and the visual system adjusts, as if preparing for dim blue depths. Fat stores build up, because the journey is long and feeding may stop or drop sharply. At the same time, parts of the digestive system can shrink while the reproductive organs develop. It is a quiet transformation that suggests one main purpose: reach the spawning grounds, then release eggs and sperm. From rivers, eels slip downstream, pass through estuaries, and enter the Atlantic like a thin ribbon of motion. After that, they become difficult to follow, because the ocean is wide and the travelers are hard to tag. For more than a century, a leading suspected destination has been the Sargasso Sea. The Sargasso Sea is not a single shore or island, but a region bounded by currents. It is known for floating mats of brown seaweed that gather into long, drifting lines. It is also a place where water masses meet and swirl, forming fronts, eddies, and gentle boundaries. And yet, despite this famous suspect, scientists do not regularly observe adult eels spawning there. They do not often capture ripe adults in spawning condition, and they rarely film a spawning event. So the mystery remains. If the Sargasso is the cradle, why is the cradle so hard to see? The evidence ladder starts with what we do find, and the first strong clue is the young. In ocean surveys, researchers collect leaf-shaped eel larvae called leptocephalus larvae. These larvae are most often found in certain zones of the Atlantic, and their sizes form a rough timeline. Smaller larvae tend to appear closer to the suspected spawning region, while larger larvae appear farther along current paths. That pattern suggests a birthplace in the open ocean, followed by a slow drift toward continents. Ocean currents can act like moving conveyor belts, carrying tiny larvae for months. For European eels, parts of the North Atlantic circulation can carry larvae eastward toward Europe and North Africa. For American eels, currents can carry larvae toward the Western Atlantic and into coastal waters. The idea is not that larvae simply float passively, because larvae can swim and adjust depth, but currents shape the broad highway, while the animal shapes small choices within that highway. As larvae approach shelves and coasts, they transform into glass eels, nearly transparent and delicate. Then they become elvers and finally yellow eels, and the inland life begins again. This life cycle is well described near coasts, which makes the missing spawning scenes feel even stranger. To reconstruct the hidden ocean years, scientists look for chemical traces inside the body. One tool is the earstones called otoliths, which can record chemical information as they grow. Otolith chemistry can reflect salinity and water chemistry, leaving a faint history of where the eel has been. Other chemical fingerprints in tissues can also hint at environmental conditions and movement between waters. These methods do not draw a clean map, but they add rungs to the evidence ladder. Another rung comes from modern tracking tags, which are smaller each year, but still a challenge for eels. Researchers attach archival tags or pop-up satellite tags and hope the device can survive depth and time. Some tagged eels reveal a striking daily rhythm during ocean travel. They often dive deep during the day, sometimes hundreds of meters down, where light is low and steady. At night, they rise into shallower layers where temperatures can be different and currents can shift. This daily up and down pattern is called eel vertical migration, and it appears in several studies. One calm explanation is safety, because deeper daytime water may reduce attention from visual hunters. Another is energy, because temperature affects metabolism, and an eel may choose layers that save fuel. A third is navigation, because different layers can offer different current directions, like changing lanes in a slow river. But here the mystery returns, because tags often fail before the final miles. Pressure can damage housings, batteries can run down, and attachment points can loosen as the animal moves. Sometimes the tag releases early, and sometimes the eel may die, leaving the final destination unknown. So we get broad routes and strong hints, but the last chapter still fades into deep water. Now, let us rest on one soft question for a moment. If you had to find one place on a moving ocean, which clue would you trust most? Smell, magnetism, or temperature? Eels may use more than one of these at once, switching depending on depth, season, and distance. One possible cue is the magnetic field, because it forms broad gradients across the Atlantic. If an eel can sense field strength or tilt, it might estimate where it sits inside the ocean basin. Another cue is smell, because the ocean carries chemical signals from plankton blooms, seafloor features, and water masses. A third cue is temperature, because the Atlantic has layers and boundaries that can act like invisible landmarks. Some scientists also consider infrasound, very low-frequency vibration from waves and currents, as a possible guide. And some consider memory of early life, where a larva experiences a water signature that later helps an adult return. Timing is another part of the puzzle, because many animals match reproduction to cycles that repeat. For eels, some hypotheses suggest timing may align with moon phase, especially darker nights. Other ideas focus on temperature layers, where a narrow band of water might be ideal for eggs and larvae. Current boundaries and frontal zones are also suspects, because they can concentrate drifting life in gentle lines. If spawning happens in a thin window, both in place and time, it becomes easy to miss even with many ships. There is also the question of depth, because spawning may occur far below the surface, beyond common sampling nets. If adults release eggs deep and briefly, cameras and nets might pass above without noticing. Eggs are small and fragile, and larvae can disperse quickly, spreading the evidence thin. And adult eels may be few, because populations have declined, making the ocean search even harder. So the mystery stays unresolved for practical reasons as much as biological ones. The Atlantic is vast, the targets are rare, and the observation windows may be short and moving. Still, the toolkit is improving in calm, steady steps. Pop-up satellite tags are getting better at pressure tolerance and data return. Autonomous gliders can patrol water columns for weeks, measuring temperature, salinity, and currents quietly. Some gliders and ships can also sample water for environmental DNA, tiny fragments shed by animals. Environmental DNA surveys may detect eel presence without capturing the animal, like hearing a whisper in water. New sampling designs can target suspected fronts and depth layers at the right season rather than sweeping broadly. Better current models can also help by tracing likely drift paths backward from larval locations. When you combine larval surveys, current models, tag data, and environmental DNA, the picture may sharpen. Researchers also compare across eel species and across years, so one unusual season does not mislead the story. Through all of this, one idea remains gentle and firm. Eels do reach a shared ocean region to reproduce. But the exact place, depth, and moment are still hard to pin down with consistent direct observation. So our evidence ladder ends, for now, with a careful gap at the top. We have strong signals from larvae, supportive clues from currents, and partial tracks from tags. We have plausible guidance cues, from magnetism to smell to temperature and rhythm. What we do not yet have, reliably, is a clear, repeated view of spawning adults in the open ocean. That is why this journey is called Vanishing, not because it is mythical, but because it is hidden in scale. In the coming years, watch for studies that combine gliders, fine-scale models, and environmental DNA along suspected boundaries. Watch for tags that last longer and report depth and temperature all the way to the end. And notice how often this mystery echoes others, where the answer may be a blend of cues rather than one key. From the hidden travel of one fish, we shift to a mystery that appears close to shore, yet is still difficult to read calmly. Imagine a shoreline at low tide, where the sea pulls back and leaves wide, shining sand. Now imagine a large whale resting there, alive, breathing, but in the wrong place. It is a scene that feels impossible, because whales are built for open water and long travel. And yet strandings happen, sometimes one animal, and sometimes many in a close cluster. We will keep an open loop here because later we will return to how groups coordinate and how one wrong cue can spread. The gentle mystery is not whether whales can swim, because they are strong swimmers. The mystery is why a strong swimmer enters shallow, risky water, where turning becomes hard. In some events, the animals are clearly sick or injured, but in others they appear outwardly healthy. Some mass strandings involve species that live in close social groups, such as pilot whales and some dolphins. In those species, staying together is normally a strength, but near shore it can become a trap. Stranding events can repeat in the same bays or coastlines over decades, as if the landscape itself matters. So the evidence ladder begins with maps and patterns, not with a single dramatic cause. If you look across records from stranding networks, you see that no one trigger explains every case. Instead, strandings seem to be a puzzle of overlapping pieces that sometimes line up at the same time. One piece is geography, because certain coastlines act like gently confusing mirrors to sound and movement. Shallow shelves, wide sandbars, and curved bays can create areas where tides shift quickly. A whale can follow a channel on the way in, then find that the channel has thinned as the tide drops. In places with large tidal ranges, the water can retreat fast enough that a group is suddenly too shallow. Some coastlines also have long, smooth beaches with few deep escape routes nearby. On a calm day, that smoothness can look safe, even though it hides a problem of depth. Another piece is prey, because whales do not wander randomly, they follow food. If schooling fish move close to shore, or if squid gather in certain shallow regions, predators may follow. In some years, prey movements align with warm currents or local blooms, bringing whales nearer to risky water. A hungry animal can make a choice that seems fine at first, then becomes dangerous as the sea changes. A third piece is health, because illness can reduce navigation, strength, or attention. Necropsies sometimes find infections, injuries, or heavy parasite loads that could weaken an animal. Hearing damage has also been reported in some cases, though not consistently across all events. But many strandings include animals that show no single obvious disease severe enough to explain the choice. That is why researchers are cautious, separating correlation from causation with each clue. The most important sense for many whales is sound, because light fades in deep water. Whales and dolphins use sound to communicate, to find prey, and to understand space. In a way, sound is their long-distance vision, and the ocean is the medium that carries it. But sound near a complex shoreline can behave in complicated ways. Reflections from the seafloor and surface can create echoes that overlap, like a crowded hallway. Changes in temperature layers can bend sound paths, making far objects seem closer or farther. In shallow water, the range of echolocation and listening can shrink, because there is less space for sound to travel cleanly. So one idea is that whales can become acoustically uncertain in certain coastal shapes. Imagine listening in a fog, where the echoes arrive from many directions with soft delay. In that kind of fog, a group following one another might keep moving even as uncertainty rises. Now add the social layer, because many mass strandings involve strong cohesion. In tightly bonded species, individuals may follow a leader or follow the group's general motion. If one animal becomes disoriented, the rest may stay with it, because separation is risky offshore. There is also helping behavior, where animals circle and remain near a struggling companion. That support can be life-saving in open water, yet near shore it can draw more bodies into the same trap. This is one reason mass strandings can appear sudden and collective, even if the first mistake was individual. It is also why rescue attempts are complicated, because animals may return to the shallows to remain together. Some strandings show repeated re-beaching after refloat, which fits the idea of social pull. That social pull is a quiet thread we will revisit later, when we talk about coordinated movement in large groups. Another proposed piece is the magnetic map we touched earlier, now expressed in a different setting. Some coastlines have local geomagnetic anomalies, small irregularities in the magnetic field caused by underlying rock. If an animal uses magnetic information as part of navigation, an anomaly could act like a misleading landmark. Some researchers have noted correlations between certain stranding locations and magnetic contours. But correlation is not proof, because coasts with complex geology can also have complex tides and prey patterns. Still, it remains a leading hypothesis in some regions, especially where strandings repeat along similar magnetic gradients. To test it, scientists compare stranding sites to magnetic maps and to nearby sites that do not strand. They also look at whether strandings happen more often during periods of geomagnetic disturbance, though results are mixed. So magnetism is a candidate piece, but not a universal key. Another piece is noise, because the modern ocean is louder than it once was in many places. Natural noise includes storms, surf, and shifting sea ice, and these can mask signals whales rely on. Human noise includes shipping, construction, and in some contexts, specific sonar activities. In certain documented events, especially involving deep diving species, strandings have been associated in time and place with sonar use. One proposed mechanism is rapid changes in diving behavior under stress, which could raise risk of injury. But again, not every stranding aligns with such sound sources, and many occur far from known sonar activity. So researchers avoid a single story and instead build a careful matrix of possibilities. Noise can be one factor among others, such as prey shifts, coastal shape, and group cohesion. It is also possible that different species respond differently to the same sound environment. Some may avoid an area, while others may continue into it if prey is present or if the group is committed to a path. Now consider weather and tides as quieter influences. Wind can push surface water toward shore, changing currents near bays and inlets. Fog and low visibility can reduce surface cues, though sound remains primary for most whales. Fast tidal changes can turn a deep channel into a shallow corridor in a short span of time. A group that enters on a rising tide may not sense how quickly the exit becomes narrow on the falling tide. In places with gently sloping beaches, a whale can be far from deep water, even when it seems only slightly shallow. This is why certain bays become known stranding sites across decades, like repeating notes in a long song. Yet, even in those places, strandings do not happen every day, which suggests the final cause is a combination. The final rung of the evidence ladder is what scientists can measure in real time, and this is where the field is growing. Better tag coverage can reveal movement patterns before a stranding, but tags are still not common enough to catch many events. Real-time acoustic monitoring with hydrophones can show changes in noise, communication, and movement. Rapid response necropsy protocols can preserve evidence, because tissues change quickly after death. Stranding networks train teams to record location, tides, weather, and body condition in consistent ways. This helps separate a thin coincidence from a repeated pattern. Even with careful work, the unresolved part remains. Because each coastline is unique, two strandings can look similar on the beach, but the underwater maps and soundscapes can be different. Events are also rare compared to daily ocean life, so the dataset grows slowly. And when a mass event does happen, it can be difficult to collect perfect measurements in time. So the mystery stays quiet and unsettled, not because people are not looking, but because the ocean is wide and the moments are brief. Here is a calm way to hold the puzzle in mind. Think of the shoreline as a gently changing map, drawn by tide, shaped by sand, and rewritten by currents. Then add a traveler who listens more than it sees and who often chooses to stay close to its companions. On most days, that traveler avoids danger with ease. But on rare days, a few small misreads can overlap, and a safe approach becomes a shallow trap. So the leading hypotheses remain a layered set. Coastal geography and tides can create physical traps that repeat in the same places. Prey movements can invite whales closer to shore at the wrong time. Illness or injury can contribute in some cases, though not all. Geomagnetic anomalies may mislead in certain regions, especially if a magnetic map is used. Social cohesion can turn one error into a group event because whales stay with each other. And acoustic interference from natural sources or human activity can add confusion in some contexts. What to watch for next is not one dramatic reveal, but better early signals. Watch for wider hydrophone networks that detect changes in soundscapes before an event. Watch for improved tags that can remain attached and report movements and dives more reliably. Watch for faster, standardized necropsy and sampling that can compare cases across countries. And watch for studies that combine magnetic maps, prey surveys, and coastline shape into one shared model. If you are listening half awake, you can rest on one gentle thought. Most strandings are not a single mystery with one answer, but a meeting point of several quiet forces. Before we continue, take a small, restful pause. If you are using this to fall asleep, we can soften the pace for a moment. Let your screen brightness drop a little until the light feels gentle. Take one slow breath in and let it out even slower. On the exhale, allow your shoulders to loosen and settle down. If your jaw is tight, let your tongue rest and leave a small space between teeth. If your hands are holding your phone, let your fingers relax their grip. If you want, tap save now, so this stays easy to find on another night. You do not have to remember anything, because the story will keep its own place. Notice the bed supporting you, steady and quiet beneath your weight. If your eyes want to close, let them close and keep listening lightly. Now we move from groups that get lost to groups that move together with astonishing grace. Think of a winter evening when the light thins and the air turns quiet. Above a field, a dark cloud gathers, but it is not weather. It is a murmuration, thousands of starlings moving as if they share one mind. The shape stretches, folds, and turns, smooth as smoke, yet made of separate birds. This is our next calm mystery. How do simple choices create such stable group motion? The puzzle is not only beauty, but control, because the group turns without a crash. There is no conductor at the front, and no single bird sees the whole pattern. Still, the flock keeps its spacing, changes direction, and stays together. You can see the same kind of unity in fish schools and in some insect swarms. A school of fish can pivot in an instant, like a single sheet of silver. A swarm of tiny insects can hover in place, drifting as one, then settling again. The motion looks effortless, but it is built from many small decisions, repeated again and again. We will keep a loop open, because later we will return to social whales and to patterns on land that also seem coordinated. For now, stay with one gentle question. How can local rules, followed by neighbors, produce a global shape that stays coherent? Scientists often begin with a simple idea. Each animal does not need a plan for the whole group. It only needs to respond to the few animals closest to it. If that response is steady, the group can self-organize into flowing order. This matters in nature because group motion can reduce danger. In a tight flock, it is harder for a predator to pick one target. In a fast-turning school, the confusion can buy a few vital seconds. Yet density has a cost because close neighbors raise the risk of collision. So the group must balance two needs: safety in numbers and space to avoid contact. That balance is one reason murmurations seem to pulse, tightening and loosening over time. It is also why the same species can form different shapes in different moments. To move up our evidence ladder, we start with careful measurement. For years, murmurations were described mostly by eye, which is charming but limited. Then researchers began using multiple synchronized cameras to capture the flock from several angles. With enough viewpoints, they can rebuild the flock in 3 dimensions. This makes it possible to measure spacing, speed, and how turns begin and spread. One striking finding is that each starling appears to track a small number of neighbors. Not every neighbor within a fixed distance, but a fixed count of nearby birds. This is called a topological rule, and it can stay stable even as density changes. If the flock compresses, each bird still follows the same number of neighbors. That can help the flock stay connected without becoming too rigid. In contrast, a purely distance-based rule can break when density changes sharply. Measurements also reveal that information moves through the flock like a wave. A turn begins in one area, then ripples across the group. It is not a shouted command and not a leader issuing directions. It is a chain of quick reactions, each bird adjusting to what it sees nearby. If you watch closely, you can sometimes see a bend travel across the cloud. This wave idea is important because it tells us what the group really is. It is not a single creature, but it behaves like a material that can transmit motion. In some analyses, the flock appears poised near a special balance point. Near that balance, small changes can spread widely without the whole flock falling apart. This may help the group stay responsive while remaining stable. But even this is not fully settled because different flocks and conditions can look different. Now we bring in the limits that keep the mystery alive. Every animal has reaction time, a small delay between seeing and moving. If the delay is too long, the wave can wobble and the group can fragment. If the response is too fast and too strong, the group can overshoot and jitter. So the flock's smoothness depends on a quiet tuning between delay and adjustment. Noise also matters because the world is messy. Wind gusts, small errors, and uneven attention all add variability. Yet the flock remains graceful, which suggests its rules are robust to small mistakes. Fish schools show a related story with a different set of senses. Fish use vision, especially in clear water, to align with nearby bodies. They also use the lateral line, a sensing system that feels water movement and pressure. The lateral line can detect the subtle wake of a neighbor, even when visibility is low. So a fish can stay in formation by feeling flow, not only by seeing shapes. This helps explain why some schools remain coordinated in dim conditions. In insects, the details vary, but local responses still dominate. Some swarms form around a visual marker like a tree or a rock, and individuals cycle through. Others form around smell cues or around airflow patterns in still evenings. The shared theme is modest decision-making paired with constant updating. Now we arrive at the modeling rung of the evidence ladder. If you want to recreate group motion, you can start with 3 calm rules. One rule is separation. Do not get too close to a neighbor. One rule is alignment. Match your direction with the neighbors you notice. One rule is attraction. Stay near the group so you are not left behind. With only these ideas, computer models can produce flock-like movement. They can form streams, arcs, and quick turns that resemble real groups. This is comforting in a way, because it shows that complexity can arise from simplicity. A few small rules, repeated thousands of times, can look like a living sculpture. But models also reveal a challenge. Many different rule sets can produce shapes that look similar from far away. So matching the appearance is not enough to prove the true rules of nature. That is why measurement matters, because it can test which models fit the real timing and spacing. For example, the speed of information waves can be compared to predicted reaction times. The neighbor number can be compared to model assumptions about attention limits. And the change in spacing under threat can be compared to model predictions about risk. Real groups also show flexibility that simple models do not always capture. A flock can change its density with light level, with wind, and with predator presence. It can also change its preferred direction relative to wind to save energy. Some studies that under stronger threat, the flock tightens and waves travel faster. Under calmer moments, spacing can widen and movement can slow slightly. This suggests the rules are not fixed like a machine. They are adjustable, like a dial the group can turn without discussing it. So we return to the core mystery in a gentler form. How does each individual choose which neighbors to watch, moment by moment? How does it switch between senses like vision and flow when conditions change? How does it avoid overload when there are bodies everywhere? One idea is that attention is limited by design, so a fixed neighbor count is practical. Another idea is that animals prioritize neighbors that are most informative, such as those moving differently. A neighbor that suddenly accelerates may attract attention more than one that stays steady. This could help a group detect change early without monitoring everyone equally. Still, this is hard to confirm because attention is not easy to measure in the wild. Now consider the most delicate open question. How do groups avoid cascading mistakes when one animal is wrong? If one bird misreads a turn, why does the whole flock not spiral into disorder? If one fish startles for no reason, why does the school not break apart every time? Part of the answer may be averaging, because many small inputs smooth one odd signal. If you follow several neighbors, one strange move may be diluted by the rest. Another part may be thresholds, where a change must be strong or shared before it spreads far. In this way, the group can stay sensitive, but not fragile. Yet, the balance between sensitivity and stability is still being worked out in detail. This is where new tools may offer calmer clarity. Some researchers are testing tiny location tags that can record movement of individuals inside groups. When these are combined with wind data and with predator observations, the context becomes clearer. Instead of guessing about threat, the study can measure when a predator is truly near. Instead of guessing about wind, it can measure gusts and turbulence along the flock's path. With enough cases, patterns may emerge about when rules shift and why. The same approach can be used in fish with underwater cameras and fine-scale flow sensors. It can also be used in insects with careful tracking in controlled light and airflow conditions. Each new dataset is like another soft rung on the evidence ladder. Here is a small, sleepy pole to hold your attention without effort. Do you find group movement more calming or more curious? You do not have to answer. Just notice the feeling and let it pass, because the feeling itself is part of why these patterns stay in our minds. There is also a practical side that reaches beyond biology. Swarm logic inspires robotics, where many small machines can coordinate without a central controller. It also informs crowd safety, where waves of motion can appear in human groups. But in nature, the purpose remains simple and old. Stay together, avoid harm, share information quickly, and keep moving with minimal waste. When we watch a murmuration, we are watching those goals expressed as shape. And we are watching how order can rise from local decisions without a master plan. So the gentle takeaway is this: complexity does not always need complicated instructions. Sometimes it needs a few calm rules repeated with steady feedback. What remains unclear is how those rules are chosen, tuned, and protected against mistakes in real time. And we will return to social influence again, because sound and culture can also travel through groups. From coordinated movement, we drift into coordinated sound, songs that spread across oceans. Imagine a calm ocean at night, where the surface looks still from above. Deep below, a humpback whale releases a long patterned song that can carry for miles. To us, it might sound like a drifting melody, but to whales it may be a social signal. This is our next gentle mystery: why whale music changes together across wide distances. Entire populations can adopt a new song pattern, like a slow cultural wave moving through water. Sometimes the change is gradual, like small edits made year by year. Other times it is sudden, like one song being replaced by another within a season. And often, these shifts happen without any obvious change in weather, food, or habitat. We will keep an open loop here. Later, we will meet a glowing ocean event seen even from space, another big pattern from small causes. We will also return to the idea of acoustic worlds, where invisible layers shape what can be heard. For now, rest with one clear question: how can changes in song spread across groups that may rarely meet face to face? Humpback whale songs are made of repeating parts arranged in a structured sequence. You can think of it as units that form phrases and phrases that form themes. Those themes are repeated in a steady order, sometimes for minutes, sometimes for hours. Over time, new units appear, old ones fade, and the whole sequence slowly shifts. When researchers record songs across years, they find both repetition and change. A familiar theme may persist, then return with a small twist, like a gentle variation. A pause may lengthen, a pitch may drift, or a new phrase may be inserted. This is what makes whale song feel alive, like a tradition that never fully settles. The surprising part is how coordinated those changes can be. In some regions, many whales sing roughly the same version within a season. Then, over a span of years, the shared version evolves as if the population is editing together. In a few well-documented cases, one song type seems to sweep in and replace another. In other places, songs blend, with mixed phrases appearing before a new shared pattern forms. So we have more than one style of change. There can be slow drift, gradual blending, or rapid replacement. Each style suggests different pathways for learning and contact. To climb our evidence ladder, we begin with recordings and timelines. For decades, researchers have used hydrophones to capture songs during migration and breeding seasons. Some recordings come from boats, some from moored instruments that listen for months at a time. Long-term listening reveals that songs have seasons with peaks during certain parts of the year. It also reveals that whales can be consistent for long stretches, then shift quickly. When recordings are compared across distant regions, patterns sometimes match in surprising ways. A phrase first heard in one area can later appear in another far away. The timing suggests that information can move through whale populations like a traveling rumor. But water is not air, and sound behaves differently below the surface. In the ocean, sound can travel extremely far, especially at certain frequencies. There are also water layers that can bend and guide sound, like gentle acoustic corridors. This means whales may hear one another at distances that would be impossible on land. So contact does not always require close physical meeting. A whale may hear a song from beyond the horizon, carried by the sea itself. That creates the possibility of indirect connection between groups. If two groups use the same migration corridor at different times, sound may still bridge the gap. If individuals move between regions, even rarely, they can also carry song features with them. This is important because whale social networks are hard to see. We cannot easily watch who spends time with whom underwater across an ocean basin, so researchers infer connection from where whales travel and what they sing. Learning is another key rung on the ladder. Humpback song appears to be learned rather than fully fixed at birth. Young whales are exposed to adult singers and patterns can be copied and practiced. Learning may continue across years, not just in early life. A whale can adjust its song to match what it hears around it, like joining a chorus. This helps explain why groups can converge on a shared song type. It also explains how novelty can spread if whales are willing to copy a new pattern. But copying is not the same as perfect duplication. Whales may reshape what they hear, smoothing it, shortening it, or blending it with older phrases. That reshaping could be why songs drift, even when everyone is listening. Now we arrive at the main hypotheses, each with calm strengths and quiet gaps. One leading hypothesis is cultural transmission. Under this view, song is a learned tradition that spreads through listening and imitation. Like a language accent, it can change as it moves, yet remain recognizable. Cultural transmission fits both slow drift and wave-like spread. It also fits the idea that whales may prefer to match local norms to be socially legible. Another hypothesis involves mate choice. In many populations, singing is most associated with males during the breeding season. One idea is that females may prefer certain patterns, or prefer novelty, or prefer stamina shown through long singing. If novelty matters, a new song could offer an advantage and spread quickly. But evidence here is mixed, because observing mate choice at sea is extremely difficult. It is hard to connect a specific song feature to actual mating outcomes with confidence. Also, whales sing in many contexts, not only in direct courtship scenes we can observe, so mate choice may contribute, but it may not be the only engine of change. A third hypothesis focuses on social signaling between males. Song may be a way to space out singers, reduce conflict, or advertise presence without physical contact. If the social landscape shifts, song patterns might shift too. For example, a dense area might favor different timing or structure than a sparse area. But again, the underwater social network is mostly hidden from our view. A fourth hypothesis involves environmental acoustics. The ocean is not a uniform stage. Background noise from wind and waves can mask some frequencies more than others. Shipping noise and other human sound can also change what is easy to hear in some regions. Water temperature and layering can shape how sound travels and which notes carry best. So a population might drift toward frequencies or rhythms that travel more clearly in its local soundscape. This idea is attractive because it links song to physics, not only to social taste. Yet it does not fully explain sudden song replacement when the environment seems similar. And it does not explain why neighboring regions with similar noise can have different songs. So environmental acoustics may tune the song, while culture shapes the pattern. Many researchers now consider blended explanations like this. Culture may drive the overall structure, while the ocean's acoustics shape the details that survive and spread. This is similar to how a message can change depending on the medium that carries it. Now we add the unresolved core. Even if whales can hear far, who hears whom and how often is still uncertain. A whale may hear a distant singer, but it may not choose to copy that singer. Copying may depend on age, on season, on social rank, or on simple opportunity. And we do not know how whales store and practice these patterns in the brain. Do they memorize whole sequences, or do they learn building blocks and rearrange them? Do they learn by repetition over weeks, or by short, intense listening windows? We also do not know how errors are handled. If one whale sings an unusual version by accident, why does the population not fragment into many dialects at once? Some stability may come from conformity, where most whales keep to the common version. Some may come from limited attention, where whales mostly copy the loudest or nearest singers. Some may come from thresholds, where a new feature only spreads if it appears in many singers. But these are still ideas more than settled facts. This makes whale song a calm frontier between biology, learning, and ocean physics. Now, what should we watch for as research continues? One approach is to combine tagging with passive acoustics. A tag can tell where a whale travels, how deep it dives, and sometimes what it hears nearby. Meanwhile, hydrophones can map the timing and location of singers across a region. If you merge these streams, you can estimate which whales were within listening range of which songs. Researchers are also using pattern-finding methods to compare huge libraries of recordings. This can reveal subtle edits that a human ear might miss and clarify the true timeline of change. Better ocean measurements also help. If we know the temperature layers and currents during a season, we can model where sound likely traveled. That can test whether a wave of song change matches a wave of acoustic contact. In the best case, we begin to see the hidden social network as a shifting field of hearing. Even then, some mysteries may remain. Because song is not only a signal, it may also be a practice, a tradition, and a moving identity. A population might change its song simply because it can, and because copying is part of belonging. That is a satisfying thought for a sleepy mind. A vast ocean, and within it, a traveling melody that binds strangers into a loose community. So our gentle takeaway is this: whale songs change through learning, and those changes can spread across wide distances. Sound travel in water may allow indirect contact between groups, making cultural waves possible. Environmental conditions may shape which notes carry, while social copying shapes the pattern. What remains unresolved is the detailed pathway of transmission. Who influences whom, and why certain changes catch on. And we will keep our open loop. Later, we will meet an ocean glow that also spreads as a wide, shared pattern built from tiny causes. From sound, we move to silence, patterns on land that appear without any visible planner. Imagine a dry grassland where the wind moves softly and the ground holds its water like a secret. Now imagine that among the grasses there are bare circles, as if someone gently erased the plants in perfect patches. These are called fairy circles, and they appear in parts of Namibia and parts of Australia. They can persist for years, shift slowly, and form wide patterns that are easiest to see from above. Some circles sit alone and others cluster, repeating across a landscape like a quiet design. This is our next mystery. Why are these circles so regular, and why do they gather into orderly fields? We will keep an open loop here. We recently saw how local rules can create group motion in birds and fish. In this chapter, we will ask if similar local rules can create order in soil, water, and roots. Researchers often describe two broad camps of explanation. One camp focuses on plants and water dynamics, where vegetation organizes itself under scarcity. The other camp points to insects, especially termites, acting as ecosystem engineers. It is possible that both ideas are partly true in different places or different years. That is one reason the debate stays calm but active. Let us climb the evidence ladder, starting with what can be measured on the ground. In many fairy circle sites, the center is bare or nearly bare, while the edge has a ring of taller grasses. The ring can look healthier than the surrounding grass, as if it has an advantage. So scientists test soil moisture, soil nutrients, and root patterns in and around the circles. A common finding is that moisture patterns differ between the center and the edge. After rain, water can infiltrate and move through soil in ways that concentrate resources near the border. In some measurements, the bare center loses water differently than the vegetated ring. The ring, with roots and shade, can change evaporation and uptake in a way that stabilizes the boundary. Think of the field as a slow system of water and roots, moving in small pulses after rain. If plants compete for the same limited moisture, they can create gaps simply by drawing water away from neighbors. But if plants also help trap and store moisture near their roots, rings can form where that trapping is strongest. This is where self-organization models enter the story. Ecologists have long studied how simple feedback loops can create spots, stripes, and gaps in dry landscapes. In these models, plants locally improve conditions near themselves, for example by shading soil or slowing runoff. At the same time, plants compete for water at a wider distance, because roots spread outward. When you combine local help with longer-range competition, patterns can emerge without any planner. Under scarce water, the stable arrangement can become a patchwork of gaps and vegetation, like a living mosaic. In some simulations, you can produce circles or spots that resemble fairy circles, especially when water is limiting. This is appealing because it uses a few calm rules repeated across space. But models are not proof, so researchers look for fingerprints in the real world. One fingerprint is how grass responds after rain. In some sites, the ring greens up faster or stays green longer than the surrounding area. That suggests the ring may have better access to stored moisture. Another fingerprint is how circles wax and wane with climate cycles. Satellite and aerial images show that in some regions, circle visibility changes with wetter or drier periods. During certain climate phases, circles can expand, fade, or shift, rather than staying fixed forever. This supports the idea that water dynamics play a central role, at least in some places. Now we consider the insect hypothesis, which is also grounded in real observation. In several fairy circle areas, termites are present, sometimes abundant. Termites can clear vegetation, eat roots, or change soil structure in ways that affect water. Their tunnels can increase infiltration, and their activity can move nutrients and change compaction. If termites maintain a bare patch, the surrounding grasses might benefit from reduced competition for moisture. A ring could form simply because the border grasses have access to water that is not being used by plants in the center. In this view, the circle is not just a plant pattern, it is a managed space. This idea fits the fact that circles can be sharply bounded and persist for long periods. It also fits the idea of clustering, because termite colonies can be spaced in patterns across a landscape. But there is a key complication. Termite activity is present in some sites, but not always consistently detected in all circles. Some circles show signs of insects, and others do not, depending on methods and timing. Insects can also be hard to sample, because colonies may be deep and activity can shift with season. So, absence of evidence in one survey does not always mean true absence. This is one place where edge cases matter. A circle might be initiated by drought and plant competition, then later maintained by insects that use the open space. Or insects might create a patch, and then plant feedback loops sharpen and stabilize the ring. Or two regions might produce similar circles through different mechanisms, a look-alike result from different causes. This is why researchers emphasize comparing Namibia and Australia rather than merging them into one story. The soils, grasses, rainfall patterns, and insect communities can be quite different. In Namibia, many studies focus on termite links and on soil moisture dynamics around ring grasses. In Australia, some studies also investigate termites, but others emphasize plant self-organization under arid conditions. Both regions show circles, but the underlying engine may not be identical. So what do we know from more controlled fieldwork? Some teams have tried experiments that alter access to insects. For example, excluding termites from a plot or reducing their activity, then watching how circles change over time. Other teams focus on continuous moisture measurements, placing sensors in centers and edges through seasons. The long time scale is important because circles can persist for years and respond slowly. A short study can miss the gradual drift that matters most. Remote sensing helps here because satellites can track vegetation greenness across large areas and many years. With enough years, you can see whether circles respond to drought cycles and whether there are spacing changes. Some analyses suggest that circles become more pronounced under certain levels of stress, then fade when conditions ease. That behavior is consistent with self-organized patterns that appear near thresholds of water limitation. But again, termites could also respond to those same thresholds, becoming more active when conditions shift, so the signals can overlap and careful separation is needed. Now we arrive at the heart of what remains debated. If plants alone can create circles, why are some circles so crisp and long-lived? If termites maintain circles, why are circles so regular in size and spacing across wide areas? If both play roles, how do we quantify their relative contribution in each region? And how do circles begin? Because initiation may differ from maintenance. This is a common challenge in ecology. An early event may be rare, like a drought pulse or a local colony change, and then feedbacks lock the pattern in place. Once a ring exists, it can channel water and root growth in a way that keeps the boundary stable. A bare center can reduce transpiration there, allowing moisture to linger deeper down. Border grasses can reach into that moisture with long roots, gaining a quiet advantage. This concentrates growth at the edge, which further reinforces the ring. That loop, water storage and edge growth, is a simple feedback that can create order. Insects could amplify it by keeping the center bare or by changing infiltration properties. So the gentle takeaway is not that one side must win, but that patterns can be footprints of invisible feedback loops. A field can organize itself through water flow, root competition, and small engineers in the soil. The order can be real, even if no single organism intends the whole design. Now, what should we watch for next as the debate continues? Long-term experiments that exclude insects while measuring soil moisture continuously are especially valuable. If circles persist and behave the same without insects, that strengthens plant-driven explanations. If circles fade or change dramatically when insects are excluded, that strengthens the engineering explanation. Another approach is mapping root networks and soil structure in fine detail to see whether borders have consistent underground signatures. Researchers also compare circle spacing and size to rainfall gradients, testing whether patterns shift predictably with climate. More high-resolution remote sensing can track individual circles through time, revealing whether they drift, split, or merge. If circle behavior follows the mathematics of self-organized systems, that will show up in those long records. If circle behavior tracks colony boundaries or insect markers more closely, that will also emerge with better data. For now, the mystery remains softly open. We can describe the circles, measure their moisture, and model their spacing. We can find termites in many places and also find patterns that look plant-driven. But we do not yet have a single explanation that fits every circle in every region and season. So when you picture a fairy circle field, imagine a slow conversation between water and roots. Imagine the soil as a memory of rain, and the grasses as careful collectors at the edge. And in some places, imagine insects quietly reshaping the ground beneath without ever showing themselves. The landscape becomes a kind of living diagram, drawn by feedback rather than by hand. Before we go to the ocean, a short pause to keep the pacing soft. Let us pause for a minute and keep everything soft. If your eyes are heavy, you can let them rest closed. If you are still awake, you can listen like you would listen to rain. Here is one gentle question, and you can answer it in your mind without moving at all. If you could choose, would you rather solve a mystery in the sky, the sea, or the soil tonight? You do not need to pick the best one. Just notice which word feels calmer as you hear it. Sky, sea, soil. If no choice arrives, that is fine too. Your mind can float between them like a leaf on slow water. We will keep the questions optional, always. Now, let us drift out over dark water, where an entire sea can glow softly for nights at a time. Imagine a calm night at sea when the horizon is soft and the sky feels wide. Now imagine the water itself giving off a gentle, even light, as if the ocean has become a quiet lantern. This rare event is called a Milky Sea, and it is not a quick sparkle or a brief flash. It can last for hours, sometimes for nights, and reports suggest it can spread across very large areas. The puzzle is simple to say and hard to solve. Why does the glow become so widespread, so steady, and so rare? Sailors have described sailing through water that looks softly lit from within, like pale milk poured into darkness. Unlike a wake that glitters only when disturbed, a Milky Sea seems to glow whether the water is calm or moving. Some accounts describe a ship cutting a darker path, as if the glow is a thin layer that the hull gently parts. Because these events are uncommon and hard to predict, most of what we know begins as careful description. Today, a few cases have been supported by very low light satellite sensors that can detect faint nighttime glow. To understand the glow, it helps to remember that the ocean already holds many kinds of living light. Jellyfish, small crustaceans, and drifting plankton can flash when touched or stirred by waves. Those familiar flashes are often brief and patchy, like scattered stars that blink and vanish. Milky seas look different because the light is more uniform, as if the surface has been softly painted. This uniformity is one reason scientists suspect a different source than typical flashers. A leading candidate is bioluminescent bacteria, tiny cells that can make light as part of their chemistry. Unlike many larger organisms, some glowing bacteria can produce a steadier shine rather than quick sparks. But bacteria are small, and one cell is dim, so a milky sea would require an enormous crowded population. That brings us to a key concept called quorum sensing, a kind of chemical counting between bacterial cells. In quorum sensing, bacteria release molecules into the water, and they detect how strong that signal becomes. When the population is sparse, the signal stays faint, and the bacteria conserve energy by staying dark. When the population becomes dense, this signal crosses a threshold, and many cells switch on together. This makes the glow more effective, because light from a crowded community becomes visible at a distance. It is a quiet kind of coordination, like a choir that sings only when enough voices have gathered. So the mystery shifts from light to ecology. What creates a dense, surface-level crowd of glowing bacteria? One idea is that bacteria gather on organic material, such as tiny particles, slime, or drifting debris. Another idea is that they live closely with algae, especially during large blooms that change the water. Algal blooms can provide food, surfaces, and chemical conditions that bacteria may prefer. Some blooms also create sticky aggregates that clump into rafts, offering more places for bacteria to settle. Researchers sometimes connect milky seas to regions where floating material collects and lingers. Certain regions of the ocean seem to host more reports, which hints at environmental triggers that repeat. Clusters of reports in particular seasons suggest the ocean may need a specific combination of warmth and calm. Calm surface layers can matter, because strong mixing would dilute bacteria and break up their crowded zones. A stable surface layer can trap organisms near the top, holding them in the thin boundary where light escapes. Another possible factor is nutrients, because blooms and bacteria both depend on what the water can provide. Currents and eddies can create fronts, where water masses meet and collect foam, algae, and small debris. A front can act like a soft fence, keeping particles and microbes together long enough to reach high density. Satellite detections of milky seas often appear as broad, dim glows rather than sharp, bright points. When a satellite does catch an event, it can show size, location, and how long the patch persists. Those details help researchers plan, but the hardest step is arriving in time with a ship or drone. Direct sampling is rare, because an event may be far offshore and may fade before a vessel can reach it. When samples have been collected, analyses often point toward luminous bacteria as a major contributor. Still, the exact mix of species can vary, and the ocean does not offer a single simple recipe. Even if bacteria are involved, they may be living within a larger community of algae and small animals. This makes milky seas an ecosystem event, not just a single organism lighting up the water. It also raises a gentle question. Why do we see a uniform glow instead of flickering points? One answer is scale, because bacteria cover surfaces and particles so evenly that the light blends together. Another is steadiness, because bacterial light can be continuous once the chemistry is switched on. In contrast, many plankton flash only when disturbed, leaving darkness between the moments of touch. So a bacterial glow can look like a soft sheet while a flashing plankton field looks like glitter. There is also the question of why milky seas are rare, given that bacteria and algae are common. Rarity suggests that several conditions must align at once, like pieces of a slow-moving lock. You might need a large bloom, then decay that releases organic material, then calm weather that traps the layer. You might need a particular temperature range that favors the right bacterial species and their light chemistry. You might need low turbulence so the glowing community is not spread too thin to reach its quorum threshold. Even if all of this happens, a Milky Sea may still be missed unless someone is nearby to notice. This brings us to the unresolved parts, which are as practical as they are biological. We do not yet have many confirmed well-sampled events to compare, so patterns can be uncertain. We also do not always know what the water looked like beneath the glowing surface in depth and layering. A thin, glowing surface can hide a complex vertical structure of temperature, salinity, and oxygen. Those layers can change how microbes grow and how organic material accumulates. Without measurements, we must infer the hidden structure from models and from nearby observations. The species question remains open too, because different luminous bacteria may behave differently. Genomic tests on collected water could clarify which genes for light are present during an event. That is one reason future sampling is so important, because genetics can read a history that the eye cannot. Another unresolved question is how the glow sustains itself for so long without fading. Light production costs energy, so the community must have access to fuel in the form of organic material. A long-lasting glow may mean a large, ongoing supply or a highly efficient recycling loop in the patch. Others suggest that bacteria on floating aggregates may be continually fed as particles collide and merge. These ideas are plausible, but they need more direct confirmation from samples taken during peak glow. So what should we watch for as research continues, without turning the mood sharp or urgent? One hopeful step is faster satellite alerting, so ships and autonomous systems can respond quickly. Another is the use of ocean gliders and surface drones that can travel toward a detected patch on their own. With onboard sensors, they could measure temperature layers, oxygen, and the distribution of particles. They could also collect water for later lab work, capturing microbes before the glow fades. Environmental DNA methods could detect which organisms are present, even if they are hard to see directly. Better coordination between satellite teams and ocean sampling teams could turn rare sightings into studied events. Over time, a handful of well-documented Milky Seas could become a meaningful data set rather than a legend. Until then, the gentle takeaway is simple and quietly beautiful. Sometimes the planet makes light not with fire or electricity, but by letting life gather densely enough to shine. A soft glow can be a sign of invisible chemistry, tiny cells communicating in a language of molecules. And the reason it stays mysterious is not a lack of ideas, but a lack of chances to arrive at the right place in time. Now we will keep a small open loop for later, because deep sea life also hides behind brief and rare sightings. For our next transition, imagine a slow descent, as if we are leaving the glowing skin of the ocean behind. First, the surface layer, where the light looks like a pale veil spread across dark water. Then, just below the surface, where the glow fades and the sea becomes a deeper blue. Then, down toward the twilight zone, where sunlight is weak and most colors turn to gray. Then, deeper still, where pressure rises and the water feels heavy and quiet. And finally, into the darker midwater, where large animals can be present but rarely seen. From glowing surfaces, we sink deeper into the water column, where large animals can be present but rarely seen. Now we sink into the darker layers of the ocean, where sunlight thins and time feels slow. Down here, some of Earth's largest animals can live almost unseen. The giant squid is one of the most famous examples, not because it is loud, but because it is elusive. For a long time, adults were almost never observed alive in their natural habitat, and that absence created a strange kind of knowledge. We had evidence, but not a full story. We found beaks in the stomachs of deep-diving whales. We saw circular scars on sperm whales, marks that matched the pull of large suction cups. We found occasional bodies washed ashore, fragile and altered by the journey up. And very rarely, we captured a few seconds of moving life on deep cameras. From these fragments, the mind wants to build a complete scene. But the puzzle remains calm and real. How common are giant squid? Where do they breed? And how do they behave naturally? We will keep an open loop here too. Later, we will meet another mystery where rare observation keeps science uncertain even when clues are everywhere. The deep ocean limits light, time, and equipment, and that shapes what we can know. At the surface, you can search with eyes and planes and satellites. In the deep, you search with pressure-rated machines and patient listening. The water is heavy, cold, and dark, and electronics must survive for long periods without help. So our evidence ladder begins with indirect signs, then climbs slowly toward direct observation. One of the strongest indirect signs is the relationship between giant squid and sperm whales. Sperm whales dive deep and feed on large squid. When whales surface, researchers can photograph scars and compare patterns across individuals. Some scars appear as pale circles and arcs, consistent with big tentacles wrapping and pulling. The scars do not prove a specific species in every case, but they show that large squid encounters are real and physical. Beaks provide another clue. Squid beaks are tough, and they can resist digestion longer than soft tissue. When beaks are recovered, scientists can measure them and estimate the squid's size. These estimates suggest that very large squid exist and that whales take them as prey. But beaks also highlight our missing chapter. We see the end of the story in a predator's stomach, not the beginning in the squid's own life. Stranded specimens add detail, but they are difficult evidence. A squid washed ashore may be sick, injured, or altered by pressure changes as it rises. The body is soft, so it can tear or deform, making behavior hard to infer. Still, stranded animals allow scientists to study anatomy, eyes, tentacles, and reproductive organs. They also allow genetic testing, which has become an important rung on the ladder. Genetics can confirm species identity and clarify relationships across oceans. It can tell us whether animals from different regions belong to the same species or closely related ones. It can also hint at population structure, suggesting how connected different ocean basins may be. Now we climb to the newer rung, modern cameras and deep-sea landers. Deep-sea cameras can be lowered on lines, placed on the seafloor, or carried by robotic vehicles. Some are baited, using a scent source to attract deep animals into view. Baiting is useful, but it also changes behavior, because a creature drawn to food may act differently than one simply traveling. Even so, baited landers have captured occasional images of large squid, including very large individuals. These images are precious because they show posture, movement, and tentacle use in real water. In some clips, the squid approaches slowly, arms spread, as if tasting the scene with touch and scent. In others, it hovers with small fin movements, steady in the dark, not rushing, not dramatic. Each sighting is a single frame in a long film we still cannot watch in full. Another clue comes from depth and habitat preference. Giant squid are thought to spend much of their lives in deep midwater layers, not on the seafloor. Those layers are shaped by temperature, oxygen, and food availability. In many oceans, there are oxygen minimum zones, layers where oxygen is lower than above or below. Some animals avoid these zones while others exploit them, because predators may be fewer there. If giant squid prefer certain oxygen and temperature bands, that could explain why sightings cluster at certain depths. It would also explain why encounters vary by region, because ocean layering differs across basins. But the evidence is incomplete, because we do not yet have many tagged individuals to map long-term movement. Growth and lifespan are another debated area. Some studies, using hard parts like statoliths, suggest faster growth and shorter lifespans than older assumptions. Other interpretations suggest longer lives and slower development. Different methods and different species can yield different results, so estimates vary widely. This matters because breeding behavior depends on age and season, and those details shape population dynamics. Now we arrive at the missing pieces that keep the story open. One proposed behavior is vertical migration. Many midwater animals stay deep during the day and move shallower at night, following prey that rises with darkness. If giant squid do this, then they may cross depth bands on a daily rhythm. yet still remain far below human activity. This could also explain why some sightings occur at night or in deeper twilight layers rather than near the surface. Another missing piece is breeding. Where does a giant squid mate, and where are eggs released and young developed? Some squid species lay gelatinous egg masses, while others release eggs in ways that are hard to detect. If giant squid breed in specific deep zones, perhaps near particular temperature layers or current features, we may rarely sample those places. We also do not know whether breeding is seasonal or continuous in different regions. Without direct observation, reproductive behavior is inferred from anatomy of stranded specimens and from related species. So breeding remains one of the most important blank pages. Behavior is another blank page. How do giant squid hunt, and what do they hunt most often? Do they ambush, cruise, or drift and strike? How do they avoid predators besides whales? And what roles do their large eyes play in the deep? Large eyes may be useful for detecting faint light, including the glow of bioluminescent animals. In deep water, bioluminescence is common, and even a small flash can outline prey or predator. A giant eye may be a calm adaptation to a world of tiny signals. But again, we have more explanation than observation. Why is observation so rare, if the animals are real and sometimes very large? Part of the answer is encounter rate. The deep ocean is vast, and a camera sees a small window of it. Even if squid are present, they may avoid unfamiliar objects or stay outside the light cone. Lights and thrusters can disturb animals, and some deep creatures respond by retreating into darkness. Traditional submersibles can be noisy in the water, which may reduce natural encounters. So new approaches aim to be quieter and more patient. Long-duration observatories placed for months can watch without constant disturbance. Quieter camera platforms can drift rather than drive, reducing sound and vibration. Some systems use low-light sensitive cameras that require less bright illumination. All of this increases the chance of seeing natural behavior rather than startled behavior. Environmental DNA offers another promising tool. By sampling water at depth, scientists can detect traces of genetic material shed by animals. This can reveal presence even when no body is seen. If many samples are taken across layers and regions, we can map likely habitats more efficiently. Environmental DNA cannot yet tell us full behavior, but it can narrow the search window. Combined with oceanographic measurements, it may reveal which temperature and oxygen bands carry the squid's signature most often. Over time, that can guide cameras to the right layers at the right seasons. So the gentle takeaway is this: some of Earth's largest lives unfold where we almost never look. We know giant squid exist because predators bear their marks and genetics confirms their identity. Modern cameras have given us brief, quiet glimpses, but not the full daily story. What remains missing is the natural sequence of life, how they breed, how they spend their days and nights, and how common they truly are. This is not mystery for mystery's sake. It is the normal limit of human access to deep water and the cost of deep ocean logistics. Each expedition is expensive, each instrument is delicate, and each observation is a small reward for much patience. If you want to keep one calm image in mind, picture the deep ocean as a wide, dark hallway. A giant squid can pass through that hallway at any moment, but our cameras are small lamps pointed in one direction. Sometimes the animal crosses the beam and we learn a little more. Most of the time, it does not, and the hallway stays quiet. And so science moves forward by combining fragments until they begin to fit. From deep life, we return toward the surface, where gases rise from cold seas and raise new questions. Now we travel to a colder edge of the ocean, where the seafloor can seem almost alive. In some Arctic waters, instruments detect streams of bubbles rising from sediments below. They can look like the seafloor is breathing, slowly releasing gas into the water. These bubbles often contain methane, a gas with big importance for climate and chemistry. The mystery is not that methane exists here because it has many natural sources. The mystery is what controls sudden increases, and how much methane reaches the open air. We will keep an open loop because soon we will talk about ice that can speed up in surprising ways. Both stories share a theme of thresholds where small shifts can change flow. For now, let us stay calm and precise, one rung of evidence at a time. First, a simple setup, because the Arctic seafloor is layered like a quiet archive. There are sediments, buried organic matter, and in some places frozen structures of gas and water called hydrates. A methane hydrate is like a crystal cage, water holding methane under pressure and cold. If temperature rises or pressure changes, that cage can become less stable. When that happens, methane can move along pathways in the sediment and sometimes escape as bubbles. But the pathway is rarely a clean pipe straight to the surface. Instead, it is more like a maze of pores, cracks, and tiny channels. Warming water, shifting currents, and sediment properties all interact in that maze. So researchers ask two linked questions. What controls the seepage rate at the seafloor? And what controls how much of that methane survives the trip upward? To climb our evidence ladder, we start with what can be mapped in the water. One major tool is sonar, which uses sound to detect objects and layers. Bubbles reflect sound strongly, so a plume can appear as a bright column in sonar images. By running sonar lines again and again, scientists can map where plumes rise and how tall they reach. Some plume fields persist in the same place for years, like steady vents in the landscape. Others seem to vary, strengthening in one season and weakening in another. This is one reason the seafloor feels like it is breathing. The next rung is chemistry, because methane can come from different origins. Some methane is produced by microbes in shallow sediments as they break down organic material. Some methane can also rise from deeper geological sources, moving along faults. To separate these sources, researchers measure isotopes, subtle differences within methane molecules. Isotopes act like a fingerprint, suggesting whether methane is mainly biological or deeper and older. The fingerprint is not always perfect, but it adds a calmer level of detail than location alone. Sediment cores add another rung, because they let scientists see what is stored below. A core can show layers of mud and sand, old shells, and zones rich in organic matter. Cores can also reveal where hydrates occur and where methane has been trapped before. Inside cores, researchers find microbial communities adapted to low temperature and low light. Many of these microbes do not make methane, they consume it. In seafloor sediments, microbes can oxidize methane before it ever becomes a bubble. This is a natural filter, like a quiet sink that removes gas as it rises. In the water column, other microbes can also consume dissolved methane, especially where oxygen is present. So methane released at the bottom does not automatically reach the top. A large portion can dissolve into seawater, then be eaten by microbes over time. Whether that happens depends on depth, mixing, and how fast the bubbles rise. Here, depth is a key boundary layer. In deeper water, bubbles have a longer journey and more time to dissolve or be consumed. In shallower water, the path is short and more methane can vent more directly toward the surface. This is why shallow Arctic shelf seas often draw special attention. Even there, the story is not simple, because sea ice changes everything above. Sea ice acts like a lid in some seasons, reducing gas exchange between ocean and air. It also changes wind mixing, which affects how quickly dissolved methane can be brought upward. When ice is present, methane may build up in the water, then release later when ice retreats. Or it may be carried away by currents before it can reach the surface. So the surface boundary is not fixed, it changes with ice, wind, and season. This is a helpful through line for the whole video. Many mysteries happen at boundaries. River to sea for eels, sea to shore for whales, seafloor to water for methane, and soon ice to ocean for glaciers. If you are sleepy, you can simply notice that each story has a soft border where rules change. Now back to the plumes themselves. Some surveys show hundreds of seep points spread across a region, like a field of tiny vents. Yet the methane seen in water is not the same as methane reaching the air. Scientists separate these carefully because the difference matters. Measuring methane in the water can be done with water samples and sensors. Measuring methane in the air requires flux estimates and atmospheric observations. Often, plumes look dramatic in sonar, but much of the methane dissolves before it breaks the surface. In deeper areas, this is especially likely because bubbles shrink as gas leaves them. In some shallow areas, a portion can reach the surface more directly, especially during calm conditions. Even then, wind and waves can disperse gas quickly, making measurements difficult. This leads to a gentle but important point. A visible plume does not automatically mean a large atmospheric release. It means a seafloor source is active, and then many filters decide what happens next. Now, we consider what might control sudden increases in plume activity. One candidate is hydrate stability, tied to pressure and temperature. A small warming of bottom water can shift the stability zone, opening new pathways. A change in currents can bring different water masses, altering temperature and mixing near the seafloor. Another candidate is geology, because faults and cracks can act like valves. A fault can provide a route upward, and small stress changes can open or close that route. Sediment compaction and tiny landslides can also shift pathways, sometimes quietly, sometimes quickly. Another candidate is the supply of methane itself, which can vary with organic matter and microbial production. In some places, methane is being produced steadily, but only escapes when the pathway opens. In other places, deeper methane can rise when pressure gradients change, because several mechanisms can overlap. One plume field may behave differently from another. This is why researchers are cautious about broad conclusions. The Arctic is not one uniform system, but many local systems tied together by currents and ice. Now, we come to what remains unresolved and why the uncertainty persists. A major limitation is sparse winter data. In winter, ice cover and darkness make ship surveys more difficult and less frequent. Yet, winter may be when key processes occur, like methane building up under ice or currents shifting beneath. Another limitation is that many surveys are snapshots. A ship passes through, maps plumes for days, then leaves, while the system continues changing. To understand breathing, you need continuous listening. That is why year-round moorings matter. A mooring can hold sensors in place for months, recording methane, temperature, currents, and sometimes acoustic signals. Repeated surveys matter too, because they show whether plumes persist, migrate, or appear in new places. Coupled ocean-atmosphere models matter because the surface exchange depends on wind, ice, and mixing. But models need good data to stay grounded, and data are still uneven across regions. Microbes add another layer of uncertainty. Microbes can consume methane, but rates vary with oxygen, nutrients, and temperature. In some waters, methane oxidation can be fast enough to reduce concentrations quickly. In other waters, it can be slower, leaving more methane available to move upward. Even the act of bubble rise can shape microbial processing, because bubbles stir and transport. So the system is not just physics, it is biology and chemistry together. A calm way to picture it is as a series of gates. Gate 1 is release from sediment. Gate 2 is dissolution into water. Gate 3 is microbial consumption. Gate 4 is vertical transport and mixing. Gate 5 is the surface exchange, controlled by wind and ice. Different gates dominate in different places and seasons. This is why one survey can feel surprising and another can feel quiet in the same region. So what should we watch for next as the research improves? Look for more year-round moorings on shelves and slopes, especially in regions with known seep fields. Look for repeated sonar mapping, so changes can be tracked like a slow heartbeat. Look for better isotope studies that distinguish methane sources with higher confidence. Look for combined measurements, where methane in water is measured alongside direct estimates of methane in air. Look for models that include ice cover and microbial consumption together, not separately. and look for improved mapping of seafloor faults and sediment structure to test the valve idea. The gentle takeaway is that the Arctic seafloor can release methane, and some places do so in visible plumes. But what matters most for climate is how much methane escapes the water and reaches the atmosphere. In many cases, the ocean and its microbes remove much of it before it reaches the air. In some shallow areas, the pathway is shorter, and the question becomes more sensitive to season and ice. Because data are incomplete, especially in winter, uncertainty remains part of the honest story. So for now, the seafloor keeps breathing in quiet pulses, and science keeps listening in quiet patience. From deep life, we learned that rarity of observation can hide a whole biography. Here, rarity of observation can hide a whole seasonal cycle. And soon, with ice itself, we will see how flow can change when a boundary shifts. Before we move into ice itself, take another short, restful reset. Let us take a small, quiet moment to close a loop. So far, we have met a hidden compass, where animals sense direction without a map. We have met a hidden destination, where eels travel far, yet keep their spawning place mostly unseen. We have met hidden coordination, where groups move as one and songs spread through water like culture. We have met hidden patterns on land, circles shaped by slow feedback between water and roots. And we have met hidden deep worlds, where glowing seas and giant squid remind us how much happens out of view. If you are listening to drift off, you can let these ideas settle like smooth stones in a pocket. Nothing to solve right now, only a gentle sense of wonder. In a moment, we will meet hidden motion inside ice, moving faster than it looks. Now, to the cold continents, where rivers of ice flow with their own calm secrets. Imagine a glacier as a slow river of ice, flowing so steadily it feels still. Now imagine that river changing pace, not in minutes, but across months and years. In the cold continents, ice sheets spread outward toward the sea. They do not move evenly. They move through fast corridors called ice streams, like lanes of quicker flow inside a wider, slower field. Some ice streams speed up or slow down over time, sometimes surprisingly. That is our gentle mystery here. Why does a glacier suddenly move faster than it used to? The answer is not one switch. It is often a blend of water beneath the ice, the shape of the bed, and heat from the ocean. Small changes under kilometers of ice can alter the whole system above. And the reasons can shift as conditions change, so the same glacier can behave differently in different decades. We will keep an open loop as we go. Later, we will listen for Earth's steady low hum, another signal tied to moving water and ground. For now, let us stay with the ice and build the evidence ladder calmly. First, a simple picture of how ice flows. Ice deforms under its own weight, slowly bending and sliding like a very stiff fluid. But much of the speed comes not only from bending, it comes from the base where ice meets rock or sediment. If the base grips firmly, motion stays slow. If the base becomes slippery, motion can increase even if the surface looks unchanged. So we ask a quiet question. What sets the friction at the base of an ice stream? One major player is water. Water beneath ice can act like a thin layer that reduces friction. But it is not just the presence of water. It is the pressure of that water and how it connects through channels and pockets. Basal water pressure can act like a dial, turning friction up or down. The challenge is that the dial is hidden and hard to measure directly. Another major player is the bed itself. Some beds are hard rock with bumps and ridges. Others include soft sediments that can deform, almost like dense wet clay. If the bed sediments deform, the ice above can slide more easily. In that case, the resistance is not fixed. It changes as the sediment is stirred, compacted, and re-wetted. A third major player sits at the coast. Many glaciers feed into floating ice shelves, broad platforms that extend over the ocean. An ice shelf can hold back the glacier behind it, like a gentle brace. Scientists call this back pressure or buttressing. If the ice shelf thins or breaks up, that brace weakens, and the grounded ice behind can speed up. So, even though the change happens at the edge, the response can travel inland. This is one reason ocean heat matters. Warm ocean water can reach under an ice shelf and melt it from below. That melt can thin the shelf quietly, without dramatic surface signs. Over time, thinning can reduce the shelf's ability to resist flow. Then the glacier behind it can accelerate. To see these changes, researchers look from space as well as from the ice itself. Satellite radar can measure ice motion by comparing repeated images. Laser altimetry can measure changes in ice surface height, revealing thinning or thickening. With long records, scientists can watch an ice stream speed up, slow down, or shift sideways. These satellite views are like calm maps of motion, drawn in time instead of ink. On the ground, field teams place instruments that feel the glacier's small movements. GPS stations on ice can record position changes day by day. They can show seasonal pulses, where speed increases during certain periods and eases in others. Seismic sensors can detect tiny ice quakes and basal slips, hinting at movement at the bed. Radar surveys can image internal layers and sometimes reveal bed conditions indirectly. All of this helps, but direct access beneath kilometers of ice remains rare. Now we add one of the most quietly fascinating discoveries: subglacial lakes. These are lakes trapped under ice, held liquid by pressure and geothermal heat. They can fill slowly, then drain along hidden channels. When a lake drains, it can send a pulse of water along the bed. That pulse can lubricate parts of an ice stream, sometimes changing speed downstream. The effect is not always a dramatic surge. Sometimes it is a subtle change, detectable only with careful instruments. Still, it shows that the base of the ice sheet is not dry and static. It is a living hydrologic system, with storage, flow, and occasional release. Another important boundary is the grounding line. The grounding line is where ice stops resting on bedrock and begins to float as an ice shelf. If the grounding line moves inland, more ice becomes afloat, and resistance can drop. If it moves seaward, grounded ice gains contact with the bed, and resistance can increase. Grounding line position can shift with tides, with melt, and with changes in ice thickness. Because the bed beneath often slopes, grounding line movement can affect stability. In some settings, an inland sloping bed can make retreat easier once it begins. This is a feedback loop that glaciologists study carefully. Now we return to our central puzzle: sudden speedups. In many cases, speedups involve feedback loops rather than one cause. If a glacier speeds up, it can stretch and thin. Thinner ice can float more easily near the coast, changing buttressing. Thinning can also lower the surface, altering how meltwater and snowfall balance. And faster flow can deliver more ice to the ocean, affecting sea level contributions. So the system can reinforce itself, at least for a time. But glaciers can also slow down. A subglacial drainage system can evolve from a spread-out, high-pressure sheet to more efficient channels. Efficient channels can lower water pressure at the bed, increasing friction again. In that case, a seasonal speed-up can be followed by a return to slower flow. This is why some glaciers pulse rather than run away. The details depend on local geology, water supply, and the shape of the bed. Local geology matters because the bed is not smooth. Ridges, troughs, and sticky spots can anchor flow. A sticky spot is a region where friction stays high, resisting motion. If water pressure rises enough to weaken a sticky spot, speed can increase. If pressure drops, the sticky spot can regain grip. Because sticky spots are hidden, they are one reason predictions remain uncertain. Ocean conditions add another layer of complexity. Warm water does not reach every glacier the same way. Currents, sea ice, and seafloor shape control how heat is delivered to ice shelf cavities. A small change in circulation can increase melting in one place and decrease it in another. So two nearby glaciers can respond differently in the same year. This is why scientists try to couple ocean models and ice models together. If you only model ice, you miss the ocean's quiet influence. If you only model the ocean, you miss how ice geometry reshapes the cavity water flow. Better models need better data, and better data need better access. That brings us to what remains unknown. One big unknown is basal water pressure, because it is difficult to measure across wide areas. Drilling to the bed is possible, but it is expensive and local. A single borehole is like one listening point in a vast hidden cave. Conditions can vary dramatically just a few kilometers away. Another unknown is the behavior of soft sediments. Sediments can deform, drain, and strengthen in ways that depend on their grain size and water content. We have some measurements, but not enough to describe every ice stream bed. A third unknown is how subglacial channels reorganize over time. Water pathways can switch from distributed flow to channelized flow, then back again. Those switches can change friction, and therefore speed, in ways that are hard to forecast. A fourth unknown is the full shape of the bed under thick ice. Radar mapping has improved, but gaps remain and small features can matter a lot. Even a modest ridge can act like a dam, redirecting water and altering flow. Now we look toward the future tools that may soften these uncertainties. Under-ice robots are beginning to explore ice shelf cavities more directly. Improved radar imaging can reveal bed topography and internal layers with finer resolution. More year-round sensor networks can track seasonal cycles without missing the dark months. And better coupling of ocean and ice models can translate heat and melt into flow changes more realistically. As these tools improve, conclusions can change. A glacier once thought stable can be shown to be sensitive to hidden pathways. A glacier once thought to be accelerating steadily can be shown to pulse with basal water cycles. So researchers keep updating the story, not because the science is shaky, but because the sensors are getting sharper. Here is a calm way to hold the whole system in mind. Picture an ice stream as a heavy rug sliding across a floor. If the floor is dry and rough, the rug moves slowly. If a thin, wet film forms, the rug can glide. If the rug's front edge is pinned by a brace, it resists motion. If the brace thins, the rug begins to slide more freely. Under the rug, water can pool, drain, and reroute, changing friction from week to week. And the floor itself can be soft in places, yielding under the rug's weight. That is the quiet complexity beneath a surface that looks still. So the gentle takeaway is this: ice streams are fast corridors and ice sheets, and their speed can change when basal friction changes. Basal friction is controlled by water pressure, bed shape, and bed materials. Coastal ice shelves can slow glaciers, and warm ocean water can weaken that braking effect. Subglacial lakes and drainage systems can send water pulses that change motion. Feedback loops can amplify thinning and flow, but evolving water pathways can also stabilize or slow the system. What remains unknown is the detailed state of the hidden base, because direct access is limited and local geology is complex. If you want a final soft link to the next chapter, notice how this mystery is also about signals. Satellites see motion as patterns, and sensors feel small shifts underfoot. In the next mystery, we will shift from seeing to hearing. From moving ice, we listen to the planet itself, a steady vibration that never fully stops. Now we shift from what can be seen to what can be felt. Imagine standing on quiet ground at night, with no wind in the trees and no traffic nearby. It feels still. And yet, instruments say the planet is always moving, always softly vibrating. Seismometers, the same tools used to record earthquakes, detect a continuous low-frequency vibration called the Earth hum. It is present even when there are no earthquakes, like a background note that never fully stops. The frequencies are so low that you cannot hear them directly with your ears, but the sensors feel them, and the data traces them as slow waves. This is our next calm mystery. What drives this hum, and why does it change with seasons and storms? we will keep an open loop here. Later, we will meet lights in the sky that are also difficult to sense without tools. For now, listen with the idea of listening. The planet has a sound, even in apparent silence. To climb our evidence ladder, we start with the basic observation. When seismometers run continuously, they record not only earthquakes, but also constant small wiggles. At first glance, it looks like noise. But when scientists compare many sensors, patterns emerge. Some of that pattern is the Earth hum, and some is related, called microseisms. Microseisms are vibrations generated by ocean waves interacting, pushing energy into the seafloor. The Earth hum overlaps with microseisms, but it is often discussed as the broader continuous background at very low frequency. In practice, scientists analyze frequency bands and look for source relationships across time. One of the strongest correlations involves the ocean. When the ocean is rough with strong storms, the hum often grows stronger. When the ocean is calmer, the hum often weakens. This correlation appears repeatedly across long records. It suggests that the engine is not deep volcanic activity or constant tectonic grinding. Instead, it points to water and wind acting on the boundary between ocean and rock. To understand how waves can make the Earth sing, picture the ocean surface as a moving field of pressure. A wave is not just a ridge of water, it is also a moving force pressing and releasing. When waves travel, they press on the seafloor through the water column, especially in shallower regions. Even in deeper water, certain wave interactions can create pressure patterns that reach down. The most important interaction involves waves moving in different directions. When waves meet, they can form a standing pattern with alternating pressure zones. Those pressure zones can drive vertical forces on the seafloor. When the seafloor is pushed rhythmically, it sends seismic waves into the Earth. This is one way microseisms are generated, and it likely contributes to the hum as well. Some of the energy becomes surface waves that travel around the globe. Some becomes body waves that move through deeper rock. The Earth is elastic, so it can ring in response, like a very large bell made of layered stone. But unlike a bell struck once, this ringing is continually fed by wave energy. The energy is modest but constant, and because storms move and waves propagate, the sources shift in place and time. That shifting helps explain why the hum changes even though it never stops. Now we add another rung, the role of geography. Mountain ranges, ocean basins, and continental shelves shape how vibrations travel. A wave generated near one coast may travel efficiently along certain paths and fade along others. Some components of the hum appear globally coherent, meaning they show up in similar ways across distant sensors. Other components are more regional, reflecting local ocean conditions and local geology. A sensor on a stable old continent may record the hum differently than one near an active margin. Not because the hum is absent, but because the ground filters and colors the signal. This is one reason the mystery is not fully resolved. Multiple sources overlap and local geology changes how the same forcing appears in data. To sort this out, scientists use large networks of seismometers. By comparing timing, direction, and frequency content across many stations, they can estimate where energy is coming from. They also compare seismic records to ocean wave models, which predict wave height, direction, and wave-wave interactions. When predictions match observations, confidence increases. When they do not match, the gap becomes a new question. Long records add another layer of detail. Over months and years, hum strength can show seasonal rhythms. In some regions, winter storms increase wave energy and increase the hum. In other regions, seasonal sea ice changes alter wave generation and coastal coupling. Even large climate patterns can leave signatures because they shift storm tracks and wave climates. So, within what looks like noise, there are slow, readable signals. This is part of the quiet beauty of the hum. It is a background rhythm that can carry information about the ocean and atmosphere. But it is not a simple meter with one cause. Different frequency bands behave differently. Some frequencies are well explained by ocean wave interactions. Others are still debated, or only partly captured by models. Now we consider other possible contributors. Atmospheric pressure changes can push on the ground and on the sea surface. Strong winds over the ocean can generate waves, but winds can also create pressure patterns directly. Large weather systems can load and unload the crust slightly, like a gentle breathing over thousands of kilometers. These effects may contribute indirectly by shaping wave fields and perhaps directly through pressure forcing. There are also local sources of vibration, like rivers, surf, and human activity, though these tend to dominate higher frequencies. At the very low frequencies associated with the hum, the global ocean remains the strongest suspected engine. Still, separating overlapping sources is difficult, like trying to hear one instrument in an orchestra when many are playing softly at once. This is where careful signal processing becomes part of the story. Scientists use methods to separate sources based on timing, coherence, and frequency patterns. They also use machine learning tools to classify signals and distinguish storms from other background activity. These tools can help, but they must be trained and validated carefully to avoid mistaking patterns. So the field advances with both better sensors and better ways to interpret them. Now let us return to the word hum itself. The hum is not a melody with notes you could whistle. It is more like a low-pressure shimmer in the planet's body. If you could shift it into the range of hearing, it would be a very low, continuous tone and a soft, changing texture. But even without hearing it, you can picture it as a constant vibration passing through rock. The oceans, through waves, become the bow moving across the strings. The crust and mantle become the instrument. And the song is shaped by continents and basins, shelves and ridges, like the body of a violin shaping sound. This metaphor is soothing, but we keep it grounded by returning to evidence. Strength correlates with ocean activity, especially storms. The signal travels and is shaped by Earth structure. Some components are global, some local, and long records show seasonal and climate-linked variation. What remains unresolved is the exact pathway from surface waves to specific seismic modes in every context. Different coastlines may couple energy differently. Different depths and shelf shapes may amplify or damp certain frequencies. Sea ice can block waves and change coupling, altering the hum seasonally. And local geology can filter signals, complicating comparisons. This is why the hum is partly explained and partly still debated, depending on the frequency band and region. So what should we watch for next? Dense sensor networks improve location estimates and help separate local noise from global patterns. Better ocean wave models, validated against buoy and satellite data, improve predictions of wave-wave interactions. More seabed sensors, placed closer to source regions, can record signals before they are filtered by continents. And improved analysis methods can tease apart overlapping sources more reliably. As that happens, the hum may become less of a mystery and more of a measuring tool. A planetary stethoscope, listening to storms, waves, and the slow flexing of the crust. For now, the gentle takeaway is enough. The planet has a background rhythm made by water and stone together. It is always there, quiet and low, and it changes with storms and seasons. And it reminds us that even in stillness, Earth is not silent. Now we can follow one soft tease into the next chapter. If waves can make the Earth sing, what might electricity do in moist air? From vibrations in the ground, we move to a rare glow in the air, a light that appears, yet refuses to fit neatly into one explanation. Now we come to a light that is described as close and quiet, not high and distant. It is often reported during storms or just after, when the air is wet and charged. Some witnesses describe a bright sphere, moving slowly, lingering longer than a normal flash. This is called ball lightning. though the name is really a label for many reports, not a single confirmed mechanism. The calm mystery is simple to say. How could a stable glowing form persist in humid air, sometimes for several seconds, sometimes longer? Ball lightning is rarely captured with reliable instruments, which is why it stays debated. Reports vary in size, color, motion, and duration. Some describe a small glow like a fist, others something larger, floating near the ground. Some describe it drifting smoothly, others describe it wobbling or moving with air currents. Some reports place it outdoors, others place it inside buildings near windows or chimneys. Across centuries and across continents, similar stories appear, but the details do not always match. That inconsistency is part of the challenge, because it can hide multiple phenomena under one name. Still, the persistence of the report suggests something real may be happening under rare conditions. We will keep an open loop here, because next we will visit upper atmosphere lights that are documented more clearly, yet still not fully understood. For now, let us stay gentle and careful, and build an evidence ladder. The first rung is the witness record. People have described slow glowing spheres near storms for a very long time. But eyewitness reports can be influenced by surprise, fear, and the difficulty of judging size and distance at night. So scientists treat them as clues, not measurements. The second rung is modern recording. In recent decades, a few videos have been offered as possible ball lightning. Some show a small luminous object moving near the ground for a short time. But interpreting a video is difficult without calibrated size, distance, and brightness. A camera can make a small close object look like a large distant one. Compression and sensor quirks can also create misleading shapes. So even videos do not automatically settle the debate. The third rung is constraint, which means asking what any explanation must satisfy. This is a calm checklist, and it helps keep the conversation grounded. First, the theory must explain energy. A glowing object needs power to shine, but it cannot usually deliver intense heat damage everywhere it goes. Second, the theory must explain lifetime. The glow, in many reports, lasts longer than an ordinary discharge. Third, the theory must explain motion. The object often drifts, sometimes against wind, sometimes along surfaces, sometimes through openings in ways that sound strange. Fourth, the theory must explain stability. A small ball of hot plasma in air should expand and fade quickly unless something holds it together or feeds it energy. Fifth, the theory must match the setting. Reports often involve storms, rain, strong electrical fields, and complex surfaces like soil, trees, wires, or buildings. So a good theory should naturally arise in those conditions, not require impossible laboratory perfection. When you hold these constraints together, you can see why ball lightning is hard to explain. Now we step into what laboratory work has achieved. Scientists can create glowing plasmas in controlled settings. They can also create luminous discharges along surfaces and through humid air. Some experiments produce small glowing blobs that persist briefly after a discharge, like a lingering ember of ionized gas. But consistent, stable, free-floating spheres are not easy to reproduce reliably. Sometimes a glow clings to an electrode or a surface rather than drifting as a detached ball. So lab results offer hints, but not a complete match to every reported detail. Now we consider leading hypotheses, each trying to fit the checklist. One family of ideas involves vaporized material, especially silicon or other aerosols created by a lightning strike. In this view, a strike hits soil, sand, or rock, and the intense heat vaporizes material. As the vapor cools, it can form tiny particles and reactive compounds that glow as they oxidize slowly. A cloud of glowing particles can appear roughly spherical, especially if air currents and electric fields shape it. Because the energy is chemical rather than purely electrical, the glow could persist for seconds. This family of ideas can also explain why reports sometimes occur near ground strikes and sandy soils. It can explain smoke-like behavior and gradual fading in some cases. But it struggles with reports where the glow appears far from any obvious strike point, or inside buildings without clear entry paths. Another family of ideas focuses on plasma, ionized air sustained by electromagnetic effects. Plasma is a state where electrons and ions move freely, allowing electrical conductivity. A storm creates strong electrical fields, and those fields can produce discharges. The challenge is confinement. Hot plasma should expand and cool quickly in open air, so plasma models often propose a structure, like a vortex or a self-organizing electromagnetic configuration that holds the plasma together. Some propose that a ball could be stabilized by a balance of electric and magnetic forces. But proving stable confinement in the messy atmosphere is difficult. So plasma ideas remain possible, but stability remains their hardest hurdle. A third set of ideas involves microwaves, sometimes called microwave cavity models. In these models, a lightning strike generates a burst of microwave energy. Under special conditions, that energy could become trapped in a cavity-like region, perhaps created by reflections or plasma boundaries. The trapped energy could then feed a luminous ball, sustaining it briefly. This can help with the energy and lifetime problem, because the ball is being powered from stored electromagnetic energy. But the model requires specific conditions for trapping and shaping that energy, which may be rare and hard to test. It is an elegant idea, yet difficult to confirm in the field. Another set of ideas treats the glow as a kind of corona discharge or surface discharge that appears detached due to perspective. In some situations, electrical fields around sharp objects, wires, or wet surfaces can create luminous regions. If those regions move along surfaces, they could look like a traveling ball from a distance. This could explain some indoor reports where wiring and openings might guide a discharge path. But it may not explain cases where a ball seems to float freely across open space. So again, it may describe some events rather than all. This brings us to a central reason the debate continues. Ball lightning may not be one thing. It may be a collection of rare luminous events that share a similar look. Some could be chemical aerosol glows. Some could be plasma discharges shaped by fields and airflow. Some could be electrical effects on surfaces or within buildings. And some reports could be misinterpretations of ordinary lightning afterimages or reflections. If the category includes multiple mechanisms, then inconsistent details become expected rather than suspicious. But that also means we need better observations to sort the category into clear types. Now we return to the calm checklist because it is also a retention tool for the listener. If you are drifting toward sleep, you can simply hold the checklist as a gentle rhythm: energy, lifetime, motion, stability, setting. As the theories pass by, you can see which parts they satisfy and which parts they still miss. That keeps the mind engaged without tension. Now, what should we watch for as research continues? The most useful progress would come from instrumented storm campaigns. This means setting up cameras and sensors in regions with frequent storms, waiting patiently for rare events. High-speed cameras can capture motion clearly and reduce blur. Spectral cameras can measure light wavelengths, which can hint at chemical composition. Thermal cameras can estimate heat, helping distinguish a hot plasma from a cooler chemical glow. Electric field sensors can record the local electrical environment during an event. Sound and radio sensors can detect accompanying electromagnetic signals. If many sensors record the same event, interpretation becomes stronger. Better reporting protocols also help. A standardized way to record time, location, weather conditions, and nearby strikes can reduce confusion. Even simple tools like calibrated reference objects in the field of view can help estimate size and distance. More open data sharing can allow multiple teams to analyze the same cases. Over time, this could turn a handful of ambiguous events into a meaningful dataset. Until then, honest uncertainty remains part of the story. Ball lightning sits at the intersection of physics, chemistry, and human observation. It is rare, it is brief, and it often appears when weather makes measurement difficult. The air is wet, the light is low, and attention is scattered by thunder and rain. So we have a phenomenon that many people say they have seen, but few have measured cleanly. That is a classic recipe for long debate. Still, it is a gentle debate because the goal is not to scare, but to understand. A stable glow in air would teach us something about how energy can be stored and released in storms. It would also teach us about the boundary between plasma physics and atmospheric chemistry. For now, the simplest calm statement is this. There are credible reports of a slow luminous sphere during or after storms, but reliable measurements are scarce. Any good theory must explain energy, lifetime, motion, stability, and setting without requiring extreme heat damage. Several hypothesis families exist, including slow-burning aerosol clouds, plasma configurations, and trapped electromagnetic energy. The reason it stays unresolved is rarity, observer bias, and lack of standardized multi-sensor measurements. And we keep our open loop. Next, we move upward to lights above the clouds that are better documented, yet still hold their own mysteries. Before the final major mystery, one more short reset to keep the ending gentle. Let us take another soft pause and keep the tone steady. If you are still listening, notice how science works in these quiet mysteries. First, a few solid constraints, the parts we can measure again and again. Then, many models, different ways to explain the same observations. Then, careful comparisons, looking for what fits without forcing it. And finally, a patient wait for better measurements, better sensors, and more time. This is not suspense. It is a calm method built for a world that does not always reveal itself quickly. If you like, you can hold a simple checklist in your mind. Observation, measurement, model, test, repeat. And if your eyes are closing, you can let the checklist fade and just listen. Now we look higher, into the thin air where storms make brief, quiet shapes of light. Now we look higher than the storm itself, into thin air most people never watch. This is the last major mystery of our long, calm collection. And the payoff is a gentle kind of wonder, not a jolt. Above some thunderstorms, brief flashes appear high in the atmosphere. They are faint and fast, often lasting less than a blink. Some are reddish and branching, called Sprites. Some are wide, expanding rings, called Elves. And some are narrow blue jets that rise from the cloud tops like quiet fountains of light. Together, these are called transient luminous events, meaning short-lived lights in the upper air. For a long time, they were reported by pilots and observers, long before good cameras confirmed them. That made them sound like stories. But once low-light cameras and satellites became sensitive enough, the stories became datasets. Now we know these events are real and not especially rare worldwide, yet a calm puzzle remains. Why do some storms produce them and others do not? And why do the shapes vary, even among storms that look similar from below? To climb our evidence ladder, we start with the simplest setup. A thunderstorm is not only rain and lightning, it is also a moving machine of charge. Inside a storm, collisions between ice particles and graupel help separate electric charge. The storm builds large regions of positive and negative charge at different heights. When the electrical field becomes strong enough, lightning can occur within the cloud or between cloud and ground. Most of what we call lightning happens inside the cloud. But sometimes the electrical effects of a lightning stroke reach upward into much thinner air. The upper atmosphere is not empty. It has layers, different densities, and different electrical properties. It is easier for electrical breakdown to occur in thinner air, but it also behaves differently. This is the quiet stage where sprites, elves, and jets appear. Sprites are often described as reddish structures above storms with branching tendrils. They usually occur high above the cloud tops in the mesosphere region. They are related to strong electrical fields that follow certain types of lightning strokes. In many studies, sprites are associated with powerful positive cloud-to-ground lightning. After such a stroke, the electric field above the storm can briefly intensify enough to trigger a discharge in the thin air. The discharge can spread quickly, forming a shape that looks like a jellyfish or a cluster of columns. The red color comes from excited nitrogen molecules emitting light in the thin atmosphere. Elves are different. An elf is not a branching shape. It is a rapidly expanding ring of light high above the storm. Elves are driven by an electromagnetic pulse from strong lightning. That pulse races upward and outward, and when it reaches the lower ionosphere, it can excite a wide region. The result is a faint, fast ring that expands over tens or hundreds of kilometers. An elf can be over before you know it happened, which is why cameras were needed to confirm it. Blue jets are different again. They rise from the tops of storms, moving upward in a narrow cone or column. They are often blue because of different emission conditions and the denser air closer to the cloud tops. Blue jets appear less common in many datasets, partly because they are harder to observe and may have different triggers. They can be obscured by cloud, and they may require specific storm structures to form. Now we move from description to observation, the next rung of our ladder. High-speed, low-light cameras have become a key tool. These cameras can record many frames per second, capturing shapes that unfold in milliseconds. Ground-based observers often point cameras at storm tops from far away, reducing cloud obstruction. Satellites have added a wider view, detecting transient luminous events across large regions. Lightning detection networks, which record the timing and location of lightning strokes, can be matched to camera events. This matching reveals patterns like which lightning polarity and strength are most associated with sprites. It also shows that not every strong lightning stroke produces an upper event. That is part of the ongoing puzzle. Storm structure matters. The height of the cloud top, the distribution of charge, and the presence of upper-level layers all influence what happens. Humidity and temperature shape the density and conductivity of air above the storm. Gravity waves, ripples in the atmosphere produced by strong storm updrafts, can also modulate conditions above. These waves can change local density and may influence where breakdown is easiest. So even above one storm, the upper air is not uniform. It is a dynamic, layered medium. This helps explain why two storms with similar lightning rates can produce different numbers of sprites or elves. Some storms may have the right charge arrangement for strong fields after a strike. Some may have lightning that is energetic but of a different polarity or geometry. Some may have cloud tops that block certain processes or alter field lines. So the statistics are rich, but not deterministic. Now we consider what is well explained and what remains unsettled. Broadly, the physics of electrical breakdown in thin air explains the existence of these events. We can model how strong fields can trigger streamer discharges in the mesosphere. We can model how lightning electromagnetic pulses can excite the ionosphere to create an elf ring. We can relate colors to excited molecules in different pressure regimes. So the big picture is not mysterious in the way it once was. But the exact triggering thresholds in real storms are still difficult to predict. This is because storms are complex and variable, and the measurements are incomplete. We rarely measure the full three-dimensional electric field around a storm at the exact moment of a sprite. We often infer conditions from lightning data and from cloud imagery, but inference has limits. Also, instruments miss many events. A camera must be pointed in the right direction, with the right exposure, and clouds can hide the view. A satellite may pass over at the wrong time, or its sensor may not be optimized for faint red events. So even our counts are incomplete, and that affects conclusions. Here is where the calm checklist style helps again. To explain why a storm produces sprites, we need to understand charge separation patterns inside the cloud. We need to know lightning polarity and strength, and how charge is removed by a stroke. We need to know the conductivity and density above the storm, which change with altitude and waves. And we need to know the timing, because fields can build and decay quickly. Each of these pieces can vary from storm to storm and from minute to minute. So uncertainty remains, not because physics is unknown, but because the system is hard to measure in full. Blue jets add another layer of unresolved detail. They seem to originate from cloud tops and may involve different discharge pathways than Sprites. Their relationship to storm dynamics is still being clarified. Some studies link jets to specific storm stages and charge configurations near the top. But observations are fewer, and the events can be partly hidden by cloud, so the data set is smaller and the conclusions are less settled. Now we return to our open loop about tools. These upper lights are hard to sense without instruments, like the earth hum and like some ocean glows. As sensors improved, the events moved from rumor to record. That is an important through line for the whole video. Many mysteries are not forever mysteries. They are mysteries until the right measurement becomes easy enough to repeat. So what should we watch for next? As research continues, coordinated networks are a big step. When many cameras observe storms from different angles, cloud blocking is reduced. When those cameras are matched to lightning networks, timing becomes precise. Satellite constellations could increase coverage, making it less likely that events are missed. Detailed storm electric field mapping, using balloons, aircraft, or advanced remote sensing, could link internal charge to upper events more directly. Better models that couple storm dynamics, lightning physics, and upper atmosphere response can reduce guesswork. Machine learning can help classify events and separate overlapping signals, but only if paired with good ground truth data. Over time, the field is moving toward prediction, not just description. But prediction will likely be probabilistic rather than absolute. A storm may have a higher chance of producing sprites under certain conditions, not a guarantee. That is realistic for a complex natural system. So the gentle takeaway is this. Even familiar storms have a quiet upper world we are still learning to measure calmly. Sprites, elves, and blue jets are real. Fast events shaped by lightning, atmospheric layers, and storm structure. We can explain the broad conditions, but we cannot always predict exactly which storms will produce them and when. The reason is a mix of missed observations, variable storms, and the difficulty of measuring electric fields at scale. And now we keep our promise of closure. This is the last major segment, and next we will tie the whole journey into one calm map of open questions. Before we end, we will take a longer, cozy recap that ties these mysteries into a single relaxing thread. Let us do a longer, soft recap, so this stays easy to replay on another night. If you want a simple way to remember the path, here is the order. Slowly. Compass, eels, strandings, swarms, songs, circles, glowing seas, deep giants, methane, ice flow, earth hum, rare glows, and lights above storms. Each one is a quiet evidence ladder, from what we observe, to what we measure, to what we still do not know. If one chapter stood out, you can quietly pick it now without moving at all. That choice can become your easy return point next time. Maybe you liked the hidden maps and animals. Maybe you liked the ocean making its own light. Maybe you liked the idea of ice moving like a river, or the planet humming under waves. There is no right answer. Just let one topic settle as your gentle favorite, like a bookmark you do not have to hold. Now we will close with a gentle summary and a calm goodbye. If you want another long, quiet, deep dive, subscribe, save this video, and let it stay in your bedtime routine. Leave one comment with the single mystery you want stretched into a full 2-hour episode, and I will build it slowly. May your night be easy and unhurried.